Okay, hi everybody. We're ready to start chapter 9, which is over microbial genetics. So let me do what I always do, start the PowerPoint up. Let's start by talking a little bit about some terms that we should be familiar with. The first of which is referred to as the organism's genome. Maybe you've heard of the Human Genome Project that was started about 10 or plus years ago, where they actually discovered all of the individual nitrogenous bases in the DNA of all of our 46 chromosomes in the human genome. So when we talk about an organism's genome, like you or me, or even a bacteria, we're talking about all of the possible genetic material found in the cells of that organism. So in you and me, it would be all of our chromosomes, all of the genes in our chromosomes. But we'd have to also remember from the endosymbiotic theory that our mitochondria and our chloroplasts in plant cells also have DNA in them as well, which is a throwback to the fact that at one time, billions of years ago, remember we said that those present-day organelles were at one time standalone prokaryotic cells. So you can go back in that chapter and read about that if you want to review that. In the case of bacteria, we have a single circular chromosome, as we'll talk about in a few minutes, but also bacteria have what are referred to as plasmids. This is extra chromosomal DNA. And I think back again in one of the earlier chapters, I think it was chapter 4, where we surveyed the prokaryotic cell, I introduced that concept of plasmids. So again, in most cells, we're talking about DNA as the primary source of genetic material for a cell, right? But remember in the virus chapter, we talked about how viruses, even though they're not cellular, they are infectious agents. That there can be DNA viruses and there can be RNA viruses. So if we're talking about a viral genome, which virologists often do, and they want to map out the genes present in that virus, you'd have to analyze, the appropriate nucleic acid, either an RNA or a DNA molecule. When we talk about chromosomes in cells, and I think you're quite familiar with that because we looked at chromosomes back in A&P, or if you took biology in high school, you heard about chromosomes. Here we have a structure which can be seen under the microscope with our microscopes in the lab, right? We can see these structures called chromosomes if they're stained. So what we're looking at is a very compressed structure composed of both DNA and associated proteins. And I'll be showing you another slide of this organization in just a few moments. But in the lower half of this slide, you can kind of see in this nematode, it looks to be, we have a nucleus, and then we can see inside this nucleus there are some chromosomes. And here, of course, is a chromosome, a replicated chromosome. You can see the two sister chromatids held together at the centromere. And if we were to delve into either of those sister chromatids in enough magnification, we have to get down really, really small in terms of our magnification, we would see the double helix here of DNA. So more on all of this coming up very soon. We mentioned a few moments ago that in bacteria, we have typically a single circular loop that comprises or defines the chromosome in a bacterial cell. While in you and I, again, we have a nucleus containing DNA. We have other organelles like mitochondria, or in the case of some plant cells, we'd have to add chloroplasts to the list because there are also extra chromosomal DNA material in both of those organelles. Back to the bacteria for just a moment. In addition to the circular chromosome, we see these small extrachromosomal circular structures, again called plasmids. And again, in RNA and DNA viruses, we can see those nucleic acids packaged in that outer protein coat. Okay, let's talk about genes. These are structures found within chromosomes. We often define a gene as a unit of hereditary material that is responsible for providing a given trait to the organism or to the cell, if it's a single-celled organism like euglena, for example, or a bacterial cell. Those genes held in that chromosome of the bacterium provide all the necessary traits that define that bacterial cell. Its ability to produce different kinds of structural proteins, its ability to produce different kinds of functional enzymes, everything that that cell has in terms of traits is defined by the material present in its genes within the chromosome or chromosomes, if we're talking about, say, you and I. So we're going to be reviewing the role that DNA plays in terms of protein synthesis very, very soon here in this lecture. But I also want to introduce this concept that in the DNA of cells, we have a variety of different kinds of genes. I mentioned one a moment ago, those that are coding for specific types of proteins. So we would talk about those particular genes that have that instruction booklet, if you will, that allows the cell to produce a given protein. We would refer to those genes as structural genes because they end up being transcribed and translated into structural entities like proteins, like enzymes that help reactions, of course, go faster. We talked about that in the previous chapter. There are other genes within cells that code for the production of RNA, which we're going to review a few minutes from now as well. This is another type of nucleic acid, but certain genes in DNA help in the synthesis of RNA. And finally, a third basic category, which we don't spend, I don't think, enough time really talking about. And these are the regulatory genes. Sometimes we talk about, certain genes getting turned on within a cell or turned off within a cell. If the gene is turned on, then the resulting protein produced as that gene is transcribed and translated is exhibited or is produced by the cell. If a gene is turned off, if you will, quote unquote, then there is no transcription and no translation. There is no protein ultimately produced by the cell. And if you simply think back to A&P when we talked about the cell cycle and how a cell, when it gets ready to divide, in an interphase, we talked about how in the S phase of interphase, the DNA replicates, and then eventually in the first stage of mitosis called prophase, the chromosomes appear, replicated chromosomes appear, and we go through those different stages of mitosis, prophase, metaphase, anaphase, telophase, with the ultimate production of the two daughter cells, if you remember, two identical daughter cells. Well, in the A&P book, they talk about a process that involves regulation of the cell cycle, and they talk about checkpoints within the cell cycle. Checkpoints are kind of like stoplights. So if the cell is given a green light, so to speak, and we'll think of that as a regulatory gene that is being expressed or turned on, then the cell can proceed beyond that checkpoint and enter into whatever the next stage is. If it gets a red light, then we can think of that as a regulatory gene that is not expressed, that is, let's say, turned off, and the cell cannot proceed beyond that checkpoint. So think of the cell cycle, think of the presence of strategic checkpoints along the way, all the way from interphase through that entire process of cell division. And so you might get a better sense of what I'm getting at here when I talk about regulation or regulatory genes. They don't really code for proteins, like structural genes do, but rather they help to regulate and govern processes, important processes within the cell. We won't talk much about regulatory genes, although we could. That gets into a pretty long, involved discussion. Very, very interesting to talk about. Another couple terms we want to sort of define, and that is the organism's genotype and its phenotype. The organism's genotype is a description of all of the genes within the cell. So for example, if you think back to biology in high school, you learned about how eye color is genetically determined, right? If you have blue eyes, we often describe that as your phenotype for eye color, either blue eyes or brown eyes. Well, if that's the physical manifestation, the eye color, what is responsible for that eye color? Well, that's what the genotype gets at. And so we often assign alleles, give them letters. Maybe you've heard of big B, or little b, often described as genotypes for brown eyes and blue eyes, respectively. I'm not going to get a lot of detail about this. This is not the place to get into this lecture. But if you've ever taken biology, principles of biology, and studied Mendelian genetics, you've learned about genotypes. You've learned about how genes are passed on in gametes and how the recombination of those genes in the organism, results in some sort of a phenotype. So I just want to define those two terms. Okay, here's that slide that I was talking about earlier that sort of just defines the DNA molecule and how it is packaged within the chromosome. So what I want to do is start with this replicated chromosome and kind of work my way through and kind of give you a better sense of what is that chromosome really composed of. If we went into ever increasing magnification, what would we see? Well, here we see again a replicated chromosomes in the metaphase stage of mitosis. Then if we zoom in on this, we would see hypercoiled, supercoiled condensed chromatin. Now this term you might remember from A&P or bio. This is the material that is inside the nucleus of a eukaryotic cell when it's not dividing. We talk about the DNA and the associated protein in a non-dividing cell in the form of chromatin. But when that cell goes to divide, the chromatin becomes very, very densely packed together, ultimately forming what you and I would recognize as a chromosome. But let's go again backwards here a little bit. We're taking this supercoiled chromatin and we're breaking it up, if you will. We're loosening it up. So here we can begin to see the individual threads shown in purple of chromatin. Here's indeed one of these little threads that we've again kind of blown up. And if we blow up one little chunk of chromatin, we can see the DNA here in purple, okay, wrapped around these histone proteins shown in pink. Okay, and if you continue to unravel the double helix here from the histone protein, then we'll eventually have what we recognize as the double helix of the molecule DNA. Okay, so kind of work your way from here all the way up through to the chromosome so you can have some sense of what a chromosome is composed of. It's again basically DNA and super highly compressed protein. Okay, DNA, deoxyribonucleic acid. You've heard about this, I'm sure. We talk about it a lot in one of the chapters in A&P. And we define it as this double helical structure composed of structural units called nucleotides, DNA nucleotides. And here we see in the lower left-hand part of the slide, a single nucleotide of DNA. What is it made up of? It's made up of 3 parts, right? This is, again, should be review. We've got a deoxyribose sugar here. It's a 5-sided pentose sugar bonded covalently to a phosphate group. And then over here, we have another structure, another component of the nucleotide called a nitrogenous base. They're just abbreviating it here, N base. And if we take these nucleotides, these DNA nucleotides, and we begin to bond them together, so we create a bond between the phosphate group of this nucleotide and the sugar of the one below it, and we do that again down here, we can begin to link these nucleotides together. Okay, we see that on the left-hand side of the double helix. We also see it on the right-hand side of the double helix. And then in the very center are the nitrogenous bases held together by hydrogen bonds. Note again that we have the base adenine opposite thymine and the base cytosine opposite guanine. So we're giving these bases obviously abbreviations, and you just simply need to remember that if you have an adenine on one side of the DNA helix, double helix, you have the thymine bonded to it via this hydrogen bond on the opposite half of the double helix. This is also shown in the little insert sketch in the upper right-hand corner here, where we're now depicting these bases, obviously with different colors. Anytime we see a yellow, we better make sure we have a green opposite it, right? And whenever we have a red, we have to have a blue opposite it. It's the same concept that we talked about right here. The sugar phosphate groups make up what's referred to as the backbone of the DNA double helix. So we have a backbone over here, the blue and gray alternating structures, and we have a backbone on the opposite side of the double helix. This is simply a review of what we just talked about. Here is the DNA molecule where we have sort of laid it out flat against the screen, sort of like a ladder in some respects, isn't it here? Where we have the rungs here in the middle, the A's and the T's and the C's and the G's. We can almost like walk up this ladder. These are the hydrogen bonds, right, that hold these bases together. And then we can see the sugar phosphate backbones here in the blue kind of ribbon structures here. So in the view in the center here, we don't obviously depict those sugar phosphate groups, but we know that they're here within the blue ribbon, and we can of course see the bases in the very center. This just gets into a little more of the chemistry, which you don't need to really worry so much about. What you need to know is the complementary base pairing here in the very center of the double helix, that these bases, the A and the T, are held together again by hydrogen bonds. The same applies for the C's and the G's. One thing that I will just briefly mention, it's not something to get all concerned about or to even worry about, I guess, but I'll just mention it. If you look at the orientation of the sugar phosphate groups here on the left side of the double helix and compare them with the right side, you might notice that the sugar phosphate groups are sort of upside down on this side as composed or compared rather to this side. And so we talk about the sugar phosphate backbone of the molecule as being anti-parallel due to the orientation of these sugar phosphate groups. Again, not something you need to really worry about. Okay, here's your first concept check question. In DNA, adenine is the complementary base for what? While cytosine is the complementary base for what? What do you think? A opposite what and C opposite what? Well, if you said C, you were correct. A opposite T and C opposite G. Okay. Not too long ago, I was talking about cell division, right? When we were discussing regulatory genes. One important part of that process of cell division occurred in interphase, when during the S phase of interphase, the cell's DNA replicated, if you remember this. Well, cells like bacteria also have to replicate through a process called binary fission. So they have to make an exact copy of their DNA so that the two newly formed bacterial cells have a full complement of all of those genes. And so let's talk a little bit about the process of DNA replication. This is a really important process. And so in essence, what's going on here is we have our, we'll call it the parent strand. Okay, so this is what's going to be found in the parent cell. It's undergoing division. Okay, this is the original parental DNA. We can see the double helix here. Remember, the sugar phosphate groups are shown as the blue ribbons. And as we've said, we have the complementary base pairing here in the very center. What has begun to happen, however, down below is the actual replication of the parent DNA. And so one of the first things that happens is the hydrogen bonds are broken between these nitrogenous bases. In fact, we can see that has happened right here and right here, hasn't it? And there's a special enzyme. It's called DNA polymerase that helps to come in and break those hydrogen bonds between the bases within the parent DNA. And as a result, we can see that the backbones begin to kind of separate from one another here, haven't they? This is called the replication fork. So now we have these two exposed parental strands of DNA that are going to act as a template for the formation of complementary DNA nucleotides that will be linked together, and we will begin to replicate our parent DNA. And that has already happened down here in the lower third or so of the sketch, where we see in red, this red ribbon is representing the sugar phosphate groups that have been linked together. And we can note that there are nitrogenous bases that have begun to combine with, or complement, I should say, the existing exposed bases of the parent DNA. So if there was a T over here in the parental DNA, we'd have to have an adenine brought in as this complementary daughter DNA. Now, you can again see how that has happened. Again, note the parent DNA was in blue, the daughter DNA is in pink here, at least the phosphate groups, sugar phosphate groups. So let's go up here. If this is a G in the parent, we're going to have a cytosine brought in as a daughter in a daughter nucleotide. If we have an adenine over here in the parent, we're going to have a thymine brought in as part of that new added daughter nucleotide. Those nucleotides will be linked together to, again, continue the formation of this daughter DNA. What base is going to be brought in here? What daughter nucleotide base? If there's a C here, we're going to have a G, right? And if there's a G over here, which there was because they were linked, remember earlier, we'd have a cytosine brought in as part of that new daughter nucleotide. Same occurs up here. We have a C, so we're going to have a G brought in here. We have a G on here. We have a C brought in over here. So this replication fork is working its way upward. The DNA polymerase is unzipping the parent DNA. It's breaking these hydrogen bonds, kind of like, you know, once they were all together like my jacket, and now I'm unzipping it, and now I have the exposed DNA bases, which will be complemented by the addition of daughter nucleotides. So when this is completed, what are we going to have? Well, we're going to have two identical double helices like we see in the lower part of this slide. Look at the bases. Compare the complementary base pairing. They're the same. They have to be. But within each of these double helices, Half of each is parent DNA in blue, and half is new daughter DNA in pink. Okay. This is called the semi-conservative process of DNA replication, because we have conserved half of that DNA in the form of parent, and half of that new DNA is daughter. So semi-conservative, we conserve some of the DNA, the parent DNA. So the result is the formation of two sets of double helices, both identical to one another, and of course identical to the parent DNA that they were formed from. Let's watch this video that talks a little bit more about DNA replication.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated, and the DNA replication machinery is assembled at the replication fork. The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semi-conservative DNA replication because half of the DNA molecule is conserved from the original molecule.
Okay. Now let's look at this process as it occurs within a bacterial cell. And you remember that in bacteria, the main chromosome is typically composed of a circular loop. Okay, so we're going to depict that here as the blue and the red. Okay. Again, in bacteria, you don't have chromosomes, like we do in our cells. We have a single double-stranded circular loop.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
Now, eventually, as you know from one of the earlier chapters, binary fission would occur within this parent cell, and eventually we would form two daughter cells, wouldn't we? And each of those two daughter cells would be genetically identical. One would get one circle loop, one would get the other chromosome. And they are, again, identical. So the cells that are formed asexually in bacteria are identical. Okay, let's again spend some time reviewing RNA, another type of nucleic acid. It too is composed of nucleotides. However, there are some differences between an RNA nucleotide and a DNA nucleotide. Nucleotides are made up of three parts: a sugar, a phosphate group, and a nitrogenous base. The phosphate group in RNA nucleotides is the same as the phosphate group in a DNA nucleotide. So that's no different. However, in RNA, the sugar is referred to as ribose sugar, in DNA, it's called deoxyribose sugar. And as the name implies, deoxy, without oxygen. This pertains to one of the carbons, one of the 5 carbons in the pentose sugar, lacking an oxygen. That's what deoxy means. While in a ribose sugar, it has an oxygen. So again, not critical to understand the chemistry of the difference chemically, but just know that they are different sugars. There's a slight modification that has occurred between the two. The other major difference pertain to the nitrogenous bases. In DNA, we just talked about the adenine and the thymine and the guanine and the cytosine. In RNA, you'll notice here, kind of hidden, we have adenine, we have guanine, we have cytosine, we do not have thymine. There is no thymine in RNA bases. It's called uracil. U-R-A-C-I-L. Sorry, it's kind of hard to see this. Uracil. There are also 3 major types of RNA molecules. There's messenger RNA, sometimes called mRNA for short. There's transfer RNA, or tRNA for short. And there's ribosomal RNA, or rRNA for short. And of course, you see in the table, they review those here. I'm not going to talk about the primer RNA. Let's talk about the 3 types of RNA and how they will work in concert with the DNA to help make a structural protein, like an enzyme. Okay. RNA, messenger RNA, is produced as a result of a process called transcription. We're going to talk about that in a few moments. But here we have the sugar phosphate backbone of messenger RNA. So it's going to be phosphate groups and ribose sugars bonded together covalently. And then in the very middle here, we have our nitrogenous bases. Now I want you to note, this is not a double helix. RNA is single-stranded. DNA is double-stranded. Okay. And again, what we're noting here in the different colors are the various letters. So the green represents G, the red or pink represents C, the blue represents U, and the orange represents A. And we're going to be talking about the linear sequence of 3 of these nitrogenous bases being defined as a codon. Codon. And those codons will later be translated into proteins. So we'll talk in just a few moments about how this messenger RNA is created, but just understand that it's single-stranded, that it is composed of sugar phosphate backbone here in orange, and that we have, again, the nitrogenous bases, adenine, uracil, cytosine, and guanine present as well. This messenger RNA is going to be created by transcribing one of the 2 strands of the DNA. So more on that a little bit later. Our second type of RNA is called transfer RNA. If you remember from A&P or bio, this particular molecule has sort of like a clover leaf shape to it. It's kind of a weird shape, but we can see if we look super close, here's some nitrogenous bases, right? Some G's and the C's, and there's a couple U's and A's, right? And there's some bonding here that helps kind of create these hairpin loops, they're called. Up here at the top, it says amino acid attachment site. So these transfer RNAs are going to do exactly what it says. They're going to transfer amino acids one at a time to the ribosome, where we're going to link those amino acids up and make the protein. That's what's going to happen in the cell. And I want you to note at the very bottom here of the molecule in dark orange, something called the anticodon region of the transfer RNA. We're going to talk more about the importance of that linear sequence of 3 bases a little bit later. But it's going to have some role to play in connecting with the codon segments in the messenger RNA. And our last type of RNA, called ribosomal RNA or rRNA, as the name sort of implies, ribosomal, ribosome. So you remember from A&P or from earlier micro that the ribosome is the organelle common to both eukaryotic and prokaryotic cells, where proteins are synthesized or assembled. So it's sort of like the factory where the car is built, as kind of a silly analogy, but it's the site for protein synthesis, basically. And here in this diagram, we can see that this ribosome is made up of what's called a large subunit, this upper big blue blob, and we have a smaller subunit in blue down below. So together they make up the ribosome. And while you don't see this blue structure is made up of RNA nucleotides. Okay, just like messenger RNA or transfer RNA was made up of phosphate groups, ribose sugars, and also those 4 different kinds of nitrogenous bases, A's and U's and C's and G's. But we depict it sort of in this sort of model form, if you will, the shape, just to kind of make it easier to understand its role. And that is, again, where the protein is going to be assembled and basically put together. Here we can see a couple of transfer RNA molecules that have fit into the ribosome. And in green, the circle and the triangle are representing individual amino acids that are going to be ultimately linked together to help in the construction of the polypeptide, which becomes the protein. And finally, we have our third RNA here called messenger. Remember us talking about that a few moments ago. It associates with the ribosome. It works with the transfer RNAs, and this is how we're going to make the protein. All right, so let's talk a little bit about the process. Let's talk about it in general, providing sort of an overview right now, and then we'll get into some more of the specifics. So what we're talking about is what occurs within a bacterial cell as the DNA in its circular chromosome is utilized to help produce some type of protein. So here we see the DNA in the chromosome. We're going to talk in just a few moments about a process called transcription. And again, I apologize for the review word kind of blocking some of this, but that's what it says, transcription of the DNA. The DNA is going to be transcribed. We're going to make messenger RNA from that. I'll show you how in just a moment. And then we will utilize these other 2 types of RNA, transfer and ribosomal, to translate that messenger RNA into ultimately a protein. Now, we're not going to get into regulatory materials or regulatory mechanisms, I should say, but that's kind of what's talked about over here. Don't worry about that for now. I do want you to know what transcription and translation refer to. And you're going to see these 2 processes at work right now. Just remember for now, transcription is where the DNA is utilized to produce messenger RNA. And then in translation, the messenger RNA is utilized with the help of transfer and ribosomal RNA to help produce a protein. Okay, and this is kind of what we just talked about. Okay. One of the first things that has to happen in our first step toward protein synthesis is this process called transcription. This is going to take place if it's a eukaryotic cell in the nucleus. If we're talking about a bacterium, we don't have a nucleus, so this is occurring in the cytoplasm. The first thing that happens is the utilization of an enzyme, which you see here in purple, called RNA polymerase. I know it's an enzyme because it ends in ASE. And what RNA polymerase is going to do is it's going to associate with a region of the DNA, specifically a region of the gene that's going to be responsible for giving us the instruction booklet, if you will, to make that given protein. And one of the first things that happens is the RNA polymerase is going to break the hydrogen bonds between the nitrogenous bases in the very center of the double helix of the DNA. And then one of the two DNA strands is going to act as a template or a mold for the formation of the messenger RNA. Now, I'm not going to get into detail with respect to the how these new RNA nucleotides are added in a particular direction. This is beyond the scope of this course, I think. But I just want you to understand that this enzyme will associate with what's called the promoter region of the gene, the beginning of the gene, and it's going to move along that entire segment of DNA to the end of that gene. And along the way, it's going to break the hydrogen bonds, and then it's going to bring in the new RNA nucleotides that will complement one of the two parent strands of DNA. And it's going to continue that process until it gets to the end. Then it'll dissociate with the DNA, and we will be left with our single-stranded messenger RNA. And then the DNA, once it's transcribed, it comes back together again as a double helix. So there's like this transcription section that moves along as the polymerase moves along the DNA. So here's what I'm trying to describe. This is a nice figure. This is in your textbook there, figure 9.13, major events in transcription. So what we have here is the DNA. Okay, so here's the parent DNA shown in blue and in pink. And the reason that we're using the two different colors, okay, is because we're going to think of the blue parent DNA as being that strand that's going to act as a template or a mold for the formation of the messenger RNA. And so here is a DNA polymerase, this big orange oval. It has associated with the promoter region. That's sort of the beginning here of the gene. And look what it's done. It has broken the hydrogen bonds, hasn't it, between the nitrogenous bases. So if there was let's say a T here, there would have been an A over here. If there had been a G here, there'd be a C here. If there had been an A here, there would have been a T here, right? This is DNA, so we have thymine. And this RNA polymerase, as I said, is going to move along the DNA until it gets to the end called the termination sequence or region, and then we will be done with transcription. But what's happening from the time we start in the promoter to the time we end at the termination sequence? That's what you have to understand. Well, as I said a moment ago, the RNA polymerase is breaking the hydrogen bonds between the two parent DNA strands. One of these two is going to act as a mold or template. And in this case, it's going to be the blue. That's why we've colored it blue. And look what's happened here. We have begun to add RNA nucleotides. Not only have we broken the hydrogen bonds between the DNA nucleotides, but the RNA polymerase is also going to assist in the bringing in of individual RNA nucleotides, which will eventually be linked together. So here's the sugar phosphate backbone here. Only here we've got ribose sugars and phosphates, right? And what do we know about the bases here? Well, they have to complement the bases of the template strand. Meaning, if I have a G here in blue, G, what's this got to be? If you said C, you were right. If we have a T here, T, thymine, what's got to be over here? If you said A, you were right. If we have a C here, cytosine, what do we have here? A G, right. What if we had an A here, adenine? What do we have here? If you said thymine, wrong, no thymine in RNA. In place of thymine, we're going to put uracil. Uracil. Okay. So you have to remember that this temporary base pair complementary sequencing going on here involves the fact that we have no thiamine in RNA. In place of thiamine, we use uracil. So don't forget that. So to review, if you have a cytosine in the DNA, you'll have a guanine here in the RNA. If you have a guanine here, you'll have a cytosine here. If you have a thiamine here, you'll have an adenine here. If you have an adenine here, you'll have a uracil in the RNA base. Now, just as quickly as the hydrogen bonds form between the DNA and the RNA nucleotides, they break as this enzyme moves along. The messenger RNA folds off to the side, and the bonds that were here and formed temporarily, they break, and the original DNA bases come back together. We reestablish the hydrogen bonds, right? So here at the bottom of the figure is the RNA polymerase getting toward the end of the gene. We have formed an ever increasingly elongated messenger RNA strand, single-stranded, right? And the DNA has folded back together again, no harm done. All it has done is it has provided the template strand to help synthesize, or in this case, transcribe, the messenger RNA. Okay, so the process of transcription, again to review, involves utilization of the DNA in the construction of a long strand of messenger RNA. The sequence of these C's and G's and A's and U's, the sequence of these is determined by what? If you said determined by the DNA, you were right. It is the DNA sequences that determine the messenger RNA sequences. Let's watch a short video on this.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Okay, very nice summary of transcription. The second part of protein synthesis is called translation. So we've talked about how the messenger RNA is transcribed from the DNA. Now we're ready to talk about the role that messenger RNA's close cousins, The transfer and the ribosomal RNAs will help in assisting the cell to make the desired end product called the protein. There are different stages to translation. You see those listed here: initiation, elongation, termination, folding, and processing of the protein. We'll talk primarily about the first 3 stages. Let's watch a couple videos of translation at work. The first one is going to talk about eukaryotic cell biology. Okay, that's obviously the title of this video. So this is what occurs in you and I right now as it pertains to translation of proteins. In prokaryotes, it's slightly different, but there's a lot of similarities. The differences are not super significant, but let's watch this process as it occurs in eukaryotic cells. To orient you, you can see at the bottom here the messenger RNA. Here's a transfer RNA carrying an amino acid. This is the anticodon sequence that we see here appearing. And then this is the first subunit of the ribosome. It's the small subunit. We're going to see the bigger subunit come in and lock into place in just a second. So we have to sort of get our players kind of lined up, ready to perform here. So let's watch this video.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site.
So I want you to note now the codon sequence here in the messenger RNA. This is a series of 3 bases. So if you remember one of the earlier slides, we had showed codons, or they had broken those up into 3 segments, 3 different base segments. So this is called the codon sequence. And here's the anticodon sequence. And what do you notice about these? They complement one another, don't they? So if we have AUG like we do here in the messenger RNA codon, the anticodon sequence of this transfer RNA must complement the codon sequence. What's opposite A? Normally it's T, but don't put down T. Why not? Because this is RNA. This is the transfer RNA molecule. So you put a U in place of that. If we have a U in the messenger RNA, which we could have, opposite it would be an A, and so on and so forth. Here's the next transfer RNA being shuttled in to the A site. Note its anticodon sequence complements the codon sequence in the messenger RNA.
The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the 3rd amino acid.
So we're basically making peptide bonds here between these amino acids as they get linked together. Remember, it's the ribosome that's shifting from left to right, okay? And what we're seeing is a process called elongation. We are elongating the polypeptide as we add the individual amino acids to it, right? It's getting longer, elongation.
The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
So the end result is the formation of this polypeptide, which again folds in a particular manner to form a protein. Let's watch this process as it occurs within a bacterial cell. Again, you'll note a lot of similarities here, but there are a few exceptions to the rule, and that really pertains to particular sites on the ribosome that I'll talk about here.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
So this is super similar, isn't it, to eukaryotic translation, with the exception of the E site in the ribosome, in the large subunit. We don't have that in eukaryotic cells. It's found in prokaryotic cells. Other than that, it's the same process. Now, this table is showing you the codon sequences within the messenger RNA and specifying which of the 20 amino acids each codon codes for. So for example, if we had the codon sequence in the messenger RNA AUG, which both of the preceding videos did, this is sometimes called the start codon, because it's where the process of translation begins. It also codes for the amino acid methionine. That would be the amino acid that would be brought in by that first transfer RNA. So you'll notice that when you take 4 bases, okay, those are adenine, uracil, cytosine, and guanine, the 4 nitrogenous bases. And when you combine them in triplets, there are 64 possible codon sequences. It's basically 4 to the 3rd power, 4 bases in triplets. There's 64 possible ways that you could combine 4 letters in triplets. there are 61 codons that code for the 20 amino acids. So you'll see that some of these amino acids, like phenylalanine, are coded for by both UUU and UUC. Both of these codons in the messenger RNA will result in phenylalanine being shuttled in by a transfer RNA. Some amino acids like leucine, valine, serine, proline, threonine, alanine, and so forth, glycine, there are 4 codon sequences, all of which will code for that specific amino acid. It will be brought in by a transfer RNA. Again, whose anticodon must complement these codon sequences. So now you can begin to see how there are many more codon sequences than there are amino acids, because you can have anywhere from 2 to 4 codons specifying a given amino acid. And I don't care if we're talking about E. coli or your liver cells in a human. All living organisms on Earth, be they prokaryotic or eukaryotic, all utilize this same universal code for translation. Pretty interesting. I want you to note the 3 stop codons here, UAA, UAG, and UGA. Those were referenced in the previous 2 videos. When those codons are in the A site of the ribosome, there's no transfer RNA that is allowed in because it doesn't code for an amino acid. It stops translation. So we're done producing the polypeptide. So you have to have a stop codon at the very end to stop translation. And again, what determined these codon sequences? Ultimately, it was the DNA, right? So we have a beginning to the gene, we have an ending to the gene, don't we? We have a beginning to the translation, we have an ending to the translation in terms of the protein production. Okay, so to summarize, we talked about how in transcription, one of the 2 parent strands is utilized as a template or a mold for the formation of messenger RNA. And in our example, we said the pink strand of the DNA was that strand that acted as a template or a mold. So the orange strand here shown as messenger RNA, the sequences of these bases came from, or complemented, I should say, the bases of the template strand of the DNA. This occurs in the nucleus of a eukaryotic cell or the cytoplasm of a prokaryotic cell. Once transcription occurs, if it's a eukaryotic cell, the messenger RNA will leave the nucleus. It goes out through one of those nuclear pores in the outer envelope of the nucleus, goes out into the cytoplasm where it associates with ribosomes, rRNA, and then also we have, of course, transfer RNAs helping to bring in those individual amino acids in translation. In translation, we have, again, the shuttling in of specific amino acids by transfer RNAs, whose anticodon sequences must complement the codon sequences of the messenger RNA. What we don't see here is the role of the ribosome, but it's here, right? That's where this process occurs. And as the ribosome shifts across the messenger RNA, as you saw in the videos, we bring in more and more transfer RNAs. We start adding more and more amino acids to our growing polypeptide chain until we get to a stop codon. So this is a kind of a nice overall summary. This is, again, another sketch to basically review what we just talked about. It's just another way of looking at it visually. It's the same thing as the previous slide. So we're talking about, again, a gene in the DNA being responsible for the ultimate production of a given protein. When we look at how a cell makes protein through this process of translation, what we would find is that there can be multiple ribosomes moving along the same messenger RNA strand. So here's the messenger RNA that's been made in transcription, okay? When it gets out of the nucleus, if it's a eukaryotic cell, and goes into the cytoplasm, or stays in the cytoplasm in the case of the prokaryotic cell, we see multiple ribosomes associating with the messenger RNA strand, starting at a beginning initiation region and then going to the end. And the result is the formation of a lot of the same protein, because you have these multiple ribosomes moving one behind the other, like we see here. There's like 7 ribosomes, isn't there? Moving along the messenger RNA. It's still being transcribed here, isn't it? But this is fully transcribed. Now it's being translated. And if we start here at the beginning of the messenger RNA strand and continue to move along it, look what happens. We start to elongate that polypeptide. Eventually, we'll get to the end of it, and we'll be done making our proteins. But the point I'm trying to make is we can have many ribosomes, many, moving along that same strand of RNA, messenger RNA, and the result is the formation of a lot of polypeptide, a lot of that same protein. And this is an amazing photograph, magnified 30,000 times, showing this polyribosomal complex in action, where we have the messenger RNA in the center here. These darker blobs are the ribosomes that are moving along it. And look at here, see these little bit lighter gray structures that are extending outwards? That's the growing polypeptide. Yeah, and it's the same protein that ultimately gets made, because you're basically, translating that same set of bases in the messenger RNA. That's pretty cool. One thing to note, that in eukaryotic transcription and translation, as it mentions here, the two processes don't occur at the same time. And the reason being that in a eukaryotic cell, you've got to have transcription taking place in the nucleus, as I said earlier, culminating in the formation of the messenger RNA, and then that leaves the nucleus, goes onto the cytoplasm, where we have translation take place. In a prokaryotic cell, you don't have a nucleus, so you can have both transcription and translation taking place almost simultaneously, sort of like we saw in that earlier 2 slides ago. Yeah, pretty interesting, or the previous slide, I should say. In eukaryotic messenger RNA, we generally code for a single protein, as I've been kind of alluding to. But what I didn't tell you is that in bacterial messenger RNA that's been transcribed, sometimes you can have more than one gene that has been transcribed. That big long messenger RNA may actually contain information that has been transcribed from several genes in a row, in a series. So that's kind of interesting. And the other interesting thing that I want to talk a little bit about is the fact that when you look at the transcription process in eukaryotic cells, I've led you to believe that once transcribed, that messenger RNA can then be translated, right? Well, I sort of forgot to mention something about that. And that's the topic of our next little discussion. And that refers to the fact that in eukaryotic transcription, there will be segments of that messenger RNA that will ultimately not be translated. So we got to do a little bit of RNA splicing here. So let's talk about that. You can read number 3, but it's not going to make much sense to you until you go to the next slide. So here is what I'm talking about. The editing of the transcribed messenger RNA prior to translation into protein. At the top of the diagram, we see the DNA template. So this is one of the 2 DNA strands. And I want you to note where it says E in kind of orange yellow, and it has I here in this kind of aquamarine cyan color, whatever that is. So we call the E segments, those are called exon segments, exon. I stands for intron segments. So what's happening? Well, we're going to transcribe the DNA, and that has happened here. So here's our messenger RNA transcript. We're noting that the exon segments of the DNA that were transcribed are still going to be called exon segments in the messenger RNA, and the same for the intron segments. If they're here in the DNA, we're going to have the complementary intron segment present in the messenger RNA. So in the same way that we have I's and E's along here, we're going to have I's and E's along the messenger RNA. But we're not going to utilize the intron regions here to help us make a protein. So we have to do a little bit of editing of the RNA. We have to splice the RNA. And there are special enzymes that are going to help us do that. And I'm not going to get into these spliceosomes, so don't worry about the terminology, but just understand what's going on. Basically, what has to happen is we have to modify the original transcript of the messenger RNA. We have to cut out the intron segments with the help of specific sorts of enzymes, and then we have to splice together the exon segments. And that has occurred right here at the bottom of the figure. We've trimmed out the intron segments with the help of special enzymes, and we've sort of re-glued, if you will, the ends of the exon segments together. And now we can translate that as normal, right? Utilizing ribosomes and transfer RNAs, like we've talked about, and we can make that polypeptide. So theoretically, you may have, I'm just going to pick a number. Let's say you've got this original mRNA transcript, the pre-edited one. Let's pretend this is 1,000 nitrogenous bases in length. We'll just pick a number, easy to kind of do the math. This may be 1,000 from beginning to end, while the post-edited exon spliced segments, this might only be 750, for example, or maybe even less than that. And you might be, of course, wondering, well, why are we taking out the intron segments and splicing the exon segments together? That would be another course to take, where you learn about what the significance of that is. So I'm not going to get into that right now. Okay, this is a good time to stop this particular lecture. I think there's probably going to be 3 segments, 3 lectures to chapter 9. It's such a large chapter. So we'll end the first one here, and in the beginning of the second lecture, we'll pick up with mutations. So get into chapter 9.